Hello, my name is Stephen Wigmore. I'm delighted to be giving the BJS lecture at the Surgical Research Society in Cambridge. And I'd like to thank Professor Mike Nicholson and Dr. Sarah Hosgood and the committee of the Surgical Research Society for inviting me to talk about future surgical innovation and the importance of regulation. The world of surgery is changing, and we are increasingly being asked to use devices such as advanced therapy medicinal products, new surgical devices, scaffolds, and other technologies are coming into surgery, such as stem cells and CRISPR. And with this becomes a requirement for us to learn about the regulation that needs to follow these technologies so that we stay on the right side of patient safety and the right side of the law. A few years ago, I was invited to chair a special inquiry into regenerative medicine research at University College London. And this was essentially around the tracheal transplant program, which had been run at UCL by Professor Maccarini, and provided some real-world examples of things going wrong. And I thought this would be an interesting place to start to discuss how people develop products which are going to be used in humans and the necessary steps for regulation. So this is the surgeon in question, Paolo Maccarini. He was an Italian surgeon who, at the time that I present the first case, was employed in Barcelona, but he was quite interesting in that he held a number of honorary contracts in other countries, including Italy, London, the Karolinska Institute later in Sweden, and he had a number of contracts in Russia as well. So the first case that we're going to discuss, it occurred in 2008, and it concerned a 36-year-old Colombian woman who was living in Barcelona, who had recurrent sepsis in her left chest because of a narrowing in her left main bronchus due to tuberculosis. And you can see that illustrated in the image below. This woman did have periods of hospitalization, but she was actually essentially quite well. And she had 2 children and held down a full-time job. She sought medical care in the Barcelona clinic, and Professor Maccarini decided that what she needed was to have a bronchial transplant. The trachea was procured in Padua and sent to Bristol, where it was processed, and it was essentially cultured in a bioreactor with cells from the mesenchymal stem cells and respiratory epithelial cells, which had been harvested from the recipient in Barcelona and also sent to Bristol. So these cells were expanded and they were incubated in a bioreactor with the trachea, which was then sent to Barcelona. The picture of the EasyJet plane just reminds me that when this case was initially presented, and in fact it was presented at the SRS meeting in the past, it was one of the sort of amusing points of the case was that EasyJet wouldn't allow the bioreactor onto their plane because it contained more than 50 mils of fluid. But perhaps the reason that they didn't want it on the plane was that they realized that there were some possible problems with regulation. So the trachea went to Barcelona, and the left main bronchus was transplanted. The woman was initially reported to do well, and the case was published only about 4 weeks after the operation in the Lancet in 2008. But unfortunately, she developed recurrent infections. Her bronchial graft completely collapsed, and although there were attempts at stenting it, it didn't really work. And so she ended up having a pneumonectomy. And she is alive and well, but she has a limited exercise tolerance, as you might expect. So let's now look at this case from a regulatory perspective. So there are actually no large animal data on this approach published, until 2 years after the transplant. Now, the implication from the protocol was that there actually had been large animal experiments, but there wasn't actually anything published on this until 2 years after the transplant. The treatment options it's been argued by a number of thoracic surgeons that they may not have been properly considered, and that actually she may have been better off having a pneumonectomy right away rather than going through this transplant procedure. There was actually no evidence that we could find during the inquiry that the trachea was procured with approval from the Italian Transplant Authority, so there's no objective evidence or paperwork for this. The lab that processed the cells and the trachea in Bristol was actually a veterinary lab. It wasn't a licensed facility under the Human Tissue Authority Act, and it wasn't also GMP compliant, so it wasn't clean or to the governance standards that you would expect for a lab producing tissue for human transplantation. The Human Tissue Authority, it turned out, were under the impression that the cells were not covered by MHRA licensing, and they also waived their regulatory right because they were under the impression that the patient was dying of esophageal cancer. Remember, I said to you that she had a chronic stricture as a secondary to tuberculosis, and there was never any suggestion of cancer. We weren't able to confirm this in writing, but the HTA wrote to us as the inquiry and claims that this was the case, and the impression that they were given. There was never any objective evidence of cell implantation into the graft, and the graft, as I mentioned, collapsed and required multiple stenting procedures before it was finally given up on. And interestingly, the Lancet refused to retract the paper despite being written to by multiple experts from around the world. So there's quite a lot that went wrong in this case. I want to introduce you to someone else now. This is a scientist, Professor Alexander Sefelian, who worked at UCL, and he had a background essentially as a nuclear physicist, and he did a PhD in cardiac imaging using isotopes. And while he was doing his PhD, he attended evening classes in polymer chemistry at London Polytechnic, and then set up a laboratory basically designing tissue-engineered products using his polymer chemistry learning. And he also ran the biomaterials regenerative medicine course at UCL. So the second case involved Alexander Sefelian, and the patient in this case was an Eritrean man who was studying in Iceland, who had a slow-growing tracheal carcinoma. Macchiarini decided that the treatment of choice should be to resect the trachea and replace the patient's own trachea with an artificial trachea seeded with stem cells. The trachea itself was made of post-PCU, which is the PCU stands for compliant polyurethane, and so it's a plastic trachea that was manufactured in the Royal Free Hospital, which is part of UCL in London. And two of these tracheas were made based on glass mandrels, which had been blown by a specialist glass blower using models from 3D reconstructed CT scans. The tracheas were transferred to the Karolinska Institute from London for seeding with undifferentiated autologous stem cells, and were transplanted after 72 hours of incubation. And it was reported to the BBC a month after the operation that the patient was doing extremely well, and that the graft had demonstrated immediate and rapid neovascularization and had an epithelial covering. And here's Paolo Macchiarini and the patient, and all looks well. And this is an immediate post-operative photograph. So again, let's have a look at this from a regulatory perspective. So again, there were no large animal data on this approach, which was in contrast to what was implied in the protocol. Again, I think several years after this transplant, there was an attempt to demonstrate its effectiveness in a rabbit model in the trachea, but there's very limited data on that. The tracheas were actually manufactured by a PhD student in a non-GMP laboratory in the Royal Free Hospital. And the European Medicines Agency and the Medicines Health Regulatory Authority were never contacted about the device. And UCLB, which is the business arm of UCL, only found out about the transplant after it had happened when they read about it in the news. The transplant protocol described undifferentiated stem cells, but actually they were bone marrow-derived peripheral blood mononuclear cells and respiratory epithelial cells, which were transplanted. And there was never any objective evidence of neovascularization or epithelialization. In fact, quite the opposite. The trachea, artificial trachea, was lined with an infected biofilm, which had multiple different organisms growing in it. Eventually, the graft became detached proximally and distally and eroded into the esophagus, and the patient died from sepsis with a permanent tracheostomy 3 years afterwards. So both of these cases are quite shocking in terms of the mistakes that were made and the lack of approval that was sought, and even the variance from the protocol that were accompanied the procedures. And Paolo Macchiarini is now in prison. He's serving a 2.5-year sentence for manslaughter in Sweden. As well as the cases that I've described, there were 4 children transplanted in London, and probably, we think, another 11 patients in other countries, predominantly Russia, were transplanted using similar technologies, and the vast majority of these patients have died and didn't survive very long after the surgery. You might think that these are just exceptional cases by very unusual people, but unfortunately that's not the case. And I'll present another example here of events that took place in a stem cell clinic in Florida, where some patients were treated for macular degeneration by injection of autologous stem cells. And the cells were injected behind the retina of both eyes. Both eyes were treated at once, and the patients were charged $5,000 per eye. Unfortunately, all of the women went blind in both eyes. And when this case was looked at, there has never been any evidence for stem cell benefit in macular degeneration. As you can understand, this is a very common eye disease, and there's a huge body of research looking into macular degeneration. Stem cells have no benefit demonstrated in any models. The actual stem cell isolation protocol was uncertain and suspect, and the treatment was unlicensed outside of a clinical trial. The stem cell clinic claims that it had been a clinical trial, which raised the question of why they charged $5,000 per eye for the women to be treated. And the blindness in these women was irreversible. So another example of bad regulation and bad outcomes for patients. So you could ask the question, why is this happening? Where does it all go wrong? And I think there are a number of different reasons. I think that the basic science is often inadequate or absent, or in the case of Macchiarini, sometimes fraudulent. There's a complete ignorance about regulatory pathways and an assumption from scientists that surgeons will deal with that, and from surgeons that scientists will deal with the regulation. So regulatory approval wasn't obtained in any of these cases, and good manufacturing processes were also not followed. And I think particularly in the case of Macchiarini, the scientific advance was driven by ego and ambition. The reason he was employed by the Karolinska Institute and why there was such a huge fallout in Sweden when the research misconduct was uncovered was that it was anticipated that he was going to win the Nobel Prize for medicine. In mitigation, it's sometimes difficult to find out about regulatory procedures and exactly what and who you need to contact about which thing. So if we look at surgery today and these currently available advanced therapy medicinal products, there are quite a lot of different options, from gene therapy to sort of tissue engineered medicines, and more coming along every day. So recently was listening to talks about putting cells into organs on perfusion with the aim of changing the characteristics of the organ or repairing damage in the organ. And these kind of modifications would all come under advanced therapy medicinal products. And the feature of all of these things is that they often require surgical replacement in precise anatomical locations to be effective. And that's where the interface comes between this innovation and surgery. And if you look at the product pathway, there are actually lots of different places where surgeons might be involved. And the chain of custody of organs or tissues or cells that are removed from a body with the intention of being used for transplantation or administration in another form is absolutely crucial, and every step of the pathway needs to be followed as the protocol says. Before Brexit, the European Medicines Agency was based in London and set the tone for the requirements for regulation of advanced therapy medicinal products and other medical devices in the UK. And after Brexit, the European Medicines Agency moved to Brussels, and the MHRA were tasked with taking over this responsibility, and they've done this in collaboration with the NHS Health Research Authority and the Human Tissue Authority, and essentially the process is almost identical, and CE marks are required for all of these things, which means that they actually have to comply with the original European Medicines Agency requirements. So in spite of Brexit, we haven't really changed at all. This is the NHS Health Research Authority front page, making the UK the easiest place in the world to do research that people can trust. That's a bold statement, if ever there was one, and one that's likely to come back and bite you. So good manufacturing practice, I'm sure you are all aware, is not just about having a clean facility to make things. It's all about the training, the competency, and the chain of custody and documentation, which follows the development and production of a device or product. And this means that it's possible to go back and examine in detail if there's a problem, what happened, and also it's a guarantee of high quality standards. So what are the barriers to adoption of good regulatory practice? So there's still a lack of clarity about some regulatory pathways, and also the bundle of evidence which is required to support a particular device or technology. There's a lack of education for surgeons. There's certainly not something which features in the medical school curriculum, or as far as I know, in the surgical training curriculum. And basic scientists also don't really know where to look for education in regulatory pathways. Some of these devices are driven by industry, and there may be competing commercial interests. And there's also a problem around this whole field in that people who call for regulation are often termed as Luddites and holding back development, which is not quite fair. So I think that there's probably the answer to this is to actually embed regulation in the research approval process and to create milestones in the development of technology which are funded, and the requirements for for progression in funding. In the UK, we have this scheme here, which the MRC run, and it's interesting that the developmental pathway funding scheme, which is sort of early stage development, is relatively easy to get funding for, and it's relatively easy to get funding for clinical trials. The bit that's really difficult is the processes needed to achieve regulation, which are large animal studies and the toxicology and safety studies that need to be done before you can embark on a trial. And I think greater attention needs to be paid to that particular requirement for development. So we've talked about cell-based and tissue-based therapies, but licensing and regulation doesn't just apply to those kind of therapies. And it can also apply to clinical decision-making algorithms. And this is another example of something that went wrong. So this is the NHS blood and transplant liver offering scheme, which was, liver allocation scheme, which was designed to address some perceived inequalities in the access to livers by individual center allocation. And the algorithm was designed by a senior statistician with a group of transplant clinicians, but there wasn't much interaction with the transplant community. And a lot of people were very interested in this and wanted to have an input, but were kind of pushed back in the development of it. The algorithm was initially only held at NHSBT, and they would not release it to the public. And essentially, there were 7 donor and 21 recipient covariates, and you would enter these covariates when you were registering a patient for transplant, and then every time a donor became available, it would be run through the database, and the allocation would be determined by the patient having the highest transplant benefit score. So almost immediately that it was released, people started to notice that there were some anomalies with it. And my colleague, Ewan Harrison, produced an online scoring system. He actually found out what the algorithm was and put an online scoring system online, on the web, so that people could actually put details in and see what their patient's transplant benefit scores were. This particular model that I'm showing here is a non-working model, which he has he's made it non-working because of concern over the regulation of it. But what was evident was that there were certain groups of patients that were being disadvantaged by the score. And the biggest area of disadvantage was for patients who had liver cancer. And there was a systematic bias against patients who had liver cancer, which is not the case in Europe or the US, where actually patients with liver cancer are prioritized. There were also some other anomalies, such as age having an inordinate impact on the likelihood of a patient receiving an organ. And so some of these things have now been addressed, and the system is working much better, but it probably could have been avoided, and the embarrassment could have been avoided if there had been wider consultation and greater field testing beforehand. It's not just because of this particular case that I've described to you, but now any decision-making or assisting devices are required by the MHRA to have approval as a Class 1 medical device. That also means that they should have appropriate indemnity arrangements. And if you look at the apps that we all have on our phones that are used for looking at MELD scores or various other scores, predictive scores, if you look in the small print of them, they all say that this is not a device to be used in patients, and it's all because of this requirement for regulation. So they're used for educational purposes only. So artificial intelligence, the final thing, this is a very tricky area, and it is also being used in the clinical decision-making space. I think artificial intelligence is definitely here to stay, and the question has been about whether you can regulate it. So particularly something which is evolving, and if you look at generative AI, the whole premise of that this is based on is that it's different every time. And so that's not something that's easy to think about how you could regulate. And AI is everywhere. It's accessible in people's bedrooms and at school. It's just everywhere. And, can you regulate something like that? And is it actually necessary to regulate it? I think probably it is, but the focus that I think probably will come to AI is that as a device that from the patient's perspective is being used to make decisions. So the actual technology and the description of the device will probably not be something that's be able to be regulated, but the use of it is probably what will be regulated. So a few final thoughts. I think that from the cases that are presented to you, although these are extreme, they do demonstrate how regulation is important for the future development of surgery. We're all likely to be involved in using devices or technologies that require regulation. And to do it properly, I think it needs to be embedded in research design. In terms of algorithms and clinical scores, just be aware if you're developing them in terms of your own research that if you want to use it in a clinical context, it does require regulatory approval and indemnity. So thank you very much for listening, and that's the end of my lecture.